will discuss different mechanism of action of different antibiotic drugs, mainly antibacterial. Also, we will talk about the mechanism of resistance that different microorganisms use to resist the activity of these antibiotic drugs. Classify antibacterial based on their target. So we have 5 mechanisms of action. First, we have antibacterials that target cell wall, mainly beta-lactams, glycopeptide, bactericin. Beta-lactams like penicillin, cephalosporins, monobactams, carbapenems. Glycopeptides, the most famous one is vancomycin. We also have other antibacterial that target plasma membrane, mainly polymyxins and lipopeptide. We also have different drugs that target the ribosomes. There are certain drugs that target 30S subunit of the ribosome, mainly aminoglycoside and tetracyclines. We also have other drugs that target the 50S subunits like macrolide, lincosamide, chloramphenicol. We also have other drugs that target certain metabolic pathway. For example, the most famous metabolic pathway is the folic acid synthesis that's targeted by a class of drugs called sulfa drugs, or example sulfonamide and sulfones. We also have these drugs that target DNA synthesis and RNA synthesis, like fluoroquinolones that target DNA synthesis and rifamycins that target the RNA synthesis. This table also summarizes the semi-classes of antibacterials, as we say, based on the mode of action and target. First class is the inhibitor of cell wall biosynthesis. These compounds target the biosynthesis of peptidoglycan, which is the primary component of the bacterial cell wall. Usually all the antibiotics or antibacterial that target the cell wall are bactericidal, because cell wall is very important for the bacteria cell. And because a human cell and different eukaryotic cell in general don't have cell wall or peptidoglycan, we consider all antibiotics that works on cell wall biosynthesis to have the optimum selective toxicity. Class of these antibacterial is the beta-lactams. Beta-lactams is a large group of compounds. The similarity between them is that all of them contain this beta-lactam ring in blue in their structure, and it's very important for the compound to have this beta-lactam ring. It's essential for activity. Any hydrolysis or opening of this ring will result in the loss of the activity. So this beta-lactam is the core structure and the most important part of the compound. We can classify beta-lactam into penicillins with this structure, cephalosporin, monobactams, carbapenem. These compounds here in the table, all of them are penicillins, and they differ in the side group R here in the structure. Penicillin G and penicillin V are the only natural penicillins. The rest are semi-synthetic. Beta-lactams target the cross-linking step of the peptidoglycan biosynthesis. So mainly peptidoglycan can be seen as these chains in the cell wall of different bacteria. We have this enzyme transpeptidase, which is the target of the beta-lactams. This enzyme works on forming these cross-linking among these different chains of peptidoglycan to make the cell wall more stable and more rigid. Glycan, as the name implies, composed of a chain of 5 amino acids with sugar. That's why they are called peptidoglycan. And we can see here we have one chain of the peptidoglycan with these the peptide chain in the atoms are in red, and we have the other peptide chain with these amino acid. The atoms are represented in green, and we can see this is the cross-linking step between the first chain of amino acid from the first peptidoglycan with the second peptide chain from the second peptidoglycan. So this is the link that penicillin or beta-lactams in general prevent the formation of this link, and this will lead to the death of the cell because this cross-linking is not happening. Or the target of the penicillin, which is the enzyme responsible of the cross-linking, is called the transpeptidase enzyme. We can see this is like the mechanism of action of the transpeptidase enzyme in the presence of its natural substrate. So this is the first peptide chain from the first peptidoglycan. We can see the serine attack on the amide bond, leading to the release of the first amino acid. In the same time, we have other amino acid from the other peptidoglycan. We can see the nitrogen attack on the carbonyl. This lead for this new or these two peptides to leave the active site of the enzyme. Here we can see the serine is free again. What's happening when we have penicillin in the active site of the enzyme? We can see the OH attack on the lactam ring, the OH of the serine in the active site. This leads to the opening of the lactam ring, but in the same time we have this ionic interaction between the COO- in the penicillin with the NH3 of the lysine in the active site of the amino acid. The ester bond at this position of the active site with the ionic bond at this position of the active site, this lead to like permanent or irreversible blockage of the active site. So the substrate cannot enter the active site. Also water cannot enter the active site to release to hydrolyze the ester bond. So this is how the penicillin block the active site of the transpeptase irreversibly, and this will prevent the reaction, the cross-linking. So we don't have cross-linking when we have penicillin in the active site. This is the same slide. In the first box here, we have, as we said, the mechanism of action of penicillin, how penicillin inhibit or block the transpeptidase active site. Some bacteria evolve to produce a new class of enzyme. We call them beta-lactamases. Beta-lactamases because they hydrolyze or inactivate penicillin. We said the most active or the most important part of the penicillin or beta-lactam in general is the beta-lactam ring. This is very essential for activity. So what beta-lactamase do is that they open the ring and they open the ring, and this will just lead to loss of activity just because we don't have the lactam ring. So this is the beta-lactamase that bacteria produce to resist and stop the activity of penicillin. We have also serine and OH in the active site of the beta-lactamases. They attack the amide ring. This results in opening of the lactam ring. Then we have a hydrogen attack from like hydrogen in the media surrounding the cells. This will release the inactivated form of the penicillin, make the enzyme free again, so the enzyme can go and attack and inactivate another molecule of penicillins or beta-lactams. So here, this is the compound, very similar to this one. It's the same exact compound. The difference is that the beta-lactam ring is opened. So this will make the compound inactive. So this is the mode of action, or this is how bacteria resist penicillin, by inactivating the compound. So here they produce enzyme that will open the ring and lead to the inactive compound. So this is the third box here, describe what scientists did to stop beta-lactamase activity. So we have this class of compounds called beta-lactamase inhibitors that scientists design. So if we look at their structure, it's very similar to the penicillin. We have the beta-lactam ring, and this side of the compound, we don't have any R or any group to the left side of the compound. But in general, these compounds work on that they interact with the beta-lactamase and form an irreversible, like covalent irreversible bond with the beta-lactamase. This will make the beta-lactamase active site very unavailable because of this new bond. So this will prevent the beta-lactamase from going and attack penicillin. So what we are doing here is just we make the beta-lactamase busy by interacting or by this reaction with the other class of compound that we designed, the beta-lactamase inhibitor. And we can see these beta-lactamase inhibitor are very effective in stopping the beta-lactamase from attacking penicillin. We can see that one molecule of beta-lactamase can bind to two different molecules of the enzyme. The second class of compound that also work on the cell wall biosynthesis of the bacteria, we have the vancomycin. Vancomycin is a member of a class of compound called the glycopeptides. All glycopeptides inhibit cell wall synthesis. They are also bactericidal because we say cell wall biosynthesis is very important for the bacterial cell. Mycin also works on preventing the cross-linking step of the peptidoglycan in the cell wall, but in different way. Vancomycin will bind to the peptide chain or the peptide part of the peptidoglycan. So they will form this cap surrounding the short peptide chain, and this will prevent the compound, or this will prevent the peptide chain from entering the active side of the transpeptidase. So we won't have this cross-linking that we would expect. However, some bacteria that are resistant to vancomycin, because they develop a way in which they modify the target of vancomycin. So this peptide chain of the peptidoglycan is the target of vancomycin. When the bacteria edit or modify this target, the vancomycin won't be able to surround or cap this peptidoglycan chain, because the bacteria insert a new amino acid instead of the 2-alanine amino acid that we can see at the end of the peptidoglycan. The resistance produce different kind of amino acid at the end of this chain, and this will stop or will prevent the vancomycin from capping, forming this cap around the peptidoglycan chain. So this is how the vancomycin work, and this is how bacteria develop resistant to vancomycin. So here the mode of resistance is that the bacteria modify the target of the antibiotic. So the antibiotic entered the cell and cannot find the target that usually it works on. That's why the bacteria become resistant to this antibiotic. This figure summarizes the mode of action of penicillin. This is the normal cross-linking step. We can see this is the transpeptidase active site. We can see the two peptide chain from the two peptide glycan. Both of them enter the active site of the transpeptidase, and we can see this new bond here in yellow that transpeptidase help to form. So the yellow bond, this is what we call cross-linking across two different chain of peptide glycan. So what penicillin do here first, it enters the active site of the transpeptidase and prevent the chains of amino acid to enter into the active site. So we don't have cross-linking. What the vancomycin does is similar, but in a different way. The vancomycin, as we say, form this cap surrounding these peptide chain, preventing them from entering the active site of the peptide glycan, leading to prevent the cross-linking step between the peptide glycan chain. Catricin is the third antibacterial that target the cell wall, but the mechanism of action is different. Catricin block the transport of a peptide glycan subunit across the cytoplasmic membrane. It's more broad spectrum compared to the penicillin or the beta-lactams and vancomycin. It targets both gram-positive and gram-negative. That the unit of the peptide glycan, the sugar, and the 5 amino acids. This unit is biosynthesized in the cytoplasm. This final unit needs to be transported across the membrane to reach the outside, where it will form the peptide glycan, where it will form the cell wall outside of on the other side of the plasma membrane. So what this drug do, it's prevent this step, transporting the peptide glycan from the cytoplasm to the outside across of the plasma membrane. Second class of antibacterial, we have the bacteria that inhibit protein synthesis by targeting ribosome. We can classify this class of compound into two classes. We have antibacterial that target the 50S subunit of the ribosome, the large subunit of the ribosome, and we have two antibacterial that target the 30S, the small subunit of the ribosome. Chloramphenicol, macrolides, and lincosamides are antibacterial that target the 50S subunit. They prevent peptide bond formation and stop protein synthesis. Aminoglycoside target the 30S ribosome subunit. They stop proofreading. This results in the incorporation of the wrong amino acid, resulting in inactive proteins. We also have tetracyclines that also target the 30S ribosomal subunit. They prevent tRNA from getting inside into the ribosome. So this also will stop and inhibit the protein synthesis, because the cell won't be able to add a new amino acid into the growing polypeptide chain, because this tRNA cannot enter to the ribosome complex. studied how different antibacterial work on bacteria, but we need also to know that different microorganisms have evolved to have different ways to avoid the effect of antibacterial. So we have here the four main resistant mechanism. So first is blocked penetration, which means the bacteria prevent the influx, like prevents the antibiotic from entering into the cell. So this way, if the antibacterial cannot enter the cell, it won't be active. So this is one way of resistant. The other way of resistant is the target modification, which means the bacteria change its target. So when the antibiotic enters the cell, it won't find the target because the target was modified or changed. So we can see this in different antibiotic. We studied one class of this resistant mechanism, target modification, in vancomycin. We saw how vancomycin, the amino acid in the peptide chain, one of the amino acid was changed. So the target of vancomycin was changed. The vancomycin cannot bind to its target because of this modification in amino acid. So this is one example on this method, target modification. The other mechanism of resistance is inactivation by enzyme. So the bacteria produce certain enzymes to inactivate, to break down, hydrolyze different antibacterial, and we took one example of this in the previous slide in beta-lactams. We saw how certain bacteria produce certain enzyme called beta-lactamase that inactivate the beta-lactams like penicillin or cephalosporin by breaking or opening the beta-lactam ring, and this will make these drugs inactive. We have the fourth mechanism of action, the efflux pump, which means the bacteria produce certain pump that will pump out the drug from inside to the outside of the cell. So there will be no time for the antibiotic to find its target and bind to it and do its activity. Instead, the bacteria will just pump it out of the cell. We have different antibiotic that the bacteria will be resistant to them, like beta-lactams, macrolide, aminoglycoside, because of this mechanism of action, which is the efflux pump. The first mechanism of action, drug modification or inactivation. So the bacteria will produce certain enzyme that can modify the antimicrobial drug, and this inactivate it or destroy it. An example is aminoglycoside. So the bacteria produce certain enzyme that makes certain modification, transfer of different chemical group, and this will lead to like a change its structure, and this will impair the binding of this drug to its target. We also, the example that we saw in previous slide, the beta-lactams. We saw that certain bacteria produce beta-lactamase to open the ring, the beta-lactam ring, and this will make the drug lose its activity. The second mechanism of resistance is the prevention of cellular uptake or efflux. So bacteria become resistant to different antibiotics by inhibiting the accumulation of the drug inside the cell. And this can happen when the cell changes its outer membrane composition. This will make it difficult for certain drugs to enter the cells or make it easier for drugs to leave the cell to the outside, and this will prevent their accumulation inside the cell. And as we said before, sometimes there is certain efflux pump that will pump the drug outside the cell. So we need to know the most important thing about this mechanism of resistance, that this is like a general way of resistance, which means one efflux pump is able to pump out different type of antimicrobial. So this will make it like more general, like one pump work on different drugs, different antibiotics. Third class, or the third mechanism of resistance, is target modification. So as we said before, every antimicrobial drug has its own target. Any change or modification on this target means the drug won't be able to bind to this target, and this will make it inactive. So this is one form of resistance, and it's very common across bacteria. The bacteria just change the structure of the target. We have, for example, some bacteria that has shared certain changes in the transpeptidase or penicillin binding protein. This is another name for the transpeptidase. So they have different active site of the transpeptidase or the PPP, which prevent the penicillin and other beta-lactams drug from entering the active site, and the drug will be inactive, and this will make the strain resistant to beta-lactams in general. Here we have different examples on drugs in which bacteria evolve resistant to this drug by doing some form of target modification. Sometimes the bacteria will change the ribosome subunit, and this will lead to a resistance on the antibiotics that work on the ribosomes, like macrolides, tetracycline, and aminoglycoside. Sometimes the bacteria will change the active site or like the structure of RNA polymerase, and this will make the enzyme, the antibiotic that works on RNA, the antibiotic we talked in the first slide, the antibiotic that target RNA synthesis like rifampin, they make it inactive because the RNA polymerase is different, the structure is different. We also have bacteria that change the active site of different metabolic pathway. So this will result in resistance to antibiotic that target this pathway. For example, the sulfa drug. And this is the example we explained in the previous slide, the vancomycin. As we said, the peptide subunit of the peptide glycan will change in structure because different amino acids are incorporated, and this will make the vancomycin inactive and make the bacteria resistant to vancomycin. We also have another example, which is target overproduction, which means the bacteria will start to produce or overproduce many of these targets. Let's imagine we have too many transpeptidase enzyme in the bacteria, so not all penicillin molecule can bind to the transpeptidase and inactivate it. So target overproduction is another example of this way of resistant target modification.